Good morning. My name is Joe Wardell, and this is Ruhi Meridia, and we are presenting on the structure and function of the yeast exosome complex.
The exosome is essential to modulate the abundance of transcripts and is the ultimate step in quality control pathways to detect and eliminate defective RNA molecules. The exosome in yeast is composed of 9 catalytically inert subunits known as XO9, a catalytically active subunit known as RRP44, and a subunit that directly contributes to long RNA binding path known as RRP6. In the cartoon illustration, the basic structure of the eukaryotic nuclear exo11 can be visualized. The left model is an apoenzyme lacking RNA with RRP44 in the open conformation, while the right image has RNA present with RRP44 in the closed conformation. The XO9 complex is composed of the barrel and cap, as shown in the blue and purple.
The goal of X-ray crystallography is to pass monochromatic X-rays through a crystal of the protein. This leads to a diffraction pattern as the X-rays are scattered in various directions by the electron density of the crystal. The constructive interference can then be utilized to determine the structure of the protein. In this experiment, the yeast exosome subunits were expressed and purified to homogeneity. The subunits were reconstituted and a his-tagged RRP6 was the final subunit to be added to form the XO11 complex. The XO11 complex was run on a histidine affinity column, and the his tag was cleaved after eluting from the column. The product was purified and incubated with a pre-annealed RNA oligonucleotide. Crystals of the protein were created via the hanging drop method, and a long needle-like crystals were formed. The crystals were flash frozen in liquid nitrogen and diffracted to a 2.8 angstrom resolution. The phase problem was solved by molecular replacement, and an electron density map was formed. The graph on the right demonstrates the process of growing protein crystals. The x-axis has time, while the main y-axis has nucleation rate, and the second y-axis has crystal growth rate. In nucleation, the molecules gather in clusters in a defined manner. In the growth phase, the nuclei have achieved critical cluster size to begin increasing in size.
The common core of eukaryotic and archaeal exosomes is a barrel-like structure of 9 polypeptides called XO9. Within XO9, 6 subunits have structural homology to RNase pH, a phosphorylytic 3' to 5' exoribonuclease. These subunits are RP41, 42, 43, 45, 46, and MTR3. The remaining 3 subunits in XO9 are the cap proteins, RP4, RP40, and CS14. The ring of the cap proteins are bound on top of the RNase pH barrel. In eukaryotic XO9, the 6 RNase pH-like subunits have amino acid changes in their active sites that render the structure catalytically inert. The active RNase RRP44, also known as DIS3, associates with XO9 to form the catalytically active XO10 complex, which is present in both the cytoplasm and nucleus of eukaryotic cells. RRP44 contains both an endoribonuclease called the PIN domain, as well as an exoribonuclease region that contains CSD1, CSD2, RMB, and S1 domains typical of RNase 2-like enzymes. In the nucleus, XO10 recruits another nuclease, RRP6, which together make up the XO11 subunit. RRP6 docks onto a conserved stretch of XO9, and the C-terminal region does not contact RNA directly. Certain cofactors are important for exosome function in specific parts of the cell. For example, helicases can disrupt RNA structure to prepare the RNA for entrance into the exosome. And once the RNA is processed by the helicase, the RNA can be threaded through the cap and barrel to be degraded in the 3' to 5' direction by the protein RRP44. The exosome can be regulated by location and function specific cofactors that associate with the 3 subunit cap. In the picture on the right, the light blue at the bottom represents the catalytic subunit of RRP44. The gray in the middle of the structure represents the 6 barrel proteins. The cap subunit is shown in orange, green, and yellow at the top. The blue subunit at the top of the structure is RRP6. The white residue is RNA, which can be seen interacting with both the 3 cap proteins as well as the RRP44.
In order to confirm that RRP44 was a catalytic subunit of the exosome, thin layer chromatography was performed to analyze the RNase activities of RRP44 in the exosome. ExoRNase assays were performed with a 3' end labeled substrate, and the assays showed that the DIS3 mutation abolished all detectable exoribonucleic activity. Addition of phosphate, magnesium ions, or both did not allow the detection of phosphorylated activity, even though under the same conditions, the archaeobacterial exosome-like complex used as a positive control was active. It can be concluded that DIS3 is the protein responsible for the in vitro exonuclease activity of the yeast exosome core. Furthermore, the DIS3 mutant phenotype suggests that this activity is very important for yeast physiology.
RNA first enters the exosome through the cap, which binds the 3' strand and threads RNA through the entrance pore by base stacking interactions and a base flipping mechanism. Positively charged amino acid residues within the exo9 barrel guide the RNA through roughly half the barrel until the RNA is channeled to the side to the same binding path that in prokaryotic exosome-like complexes and archaeal exosomes leads to a phosphorylatic site. However, in yeast, this catalytic site is absent, and RNA continues on its path to RP44, which adopts a close conformation and grabs the RNA 3' end. In the RP44, hydrolysis of the RNA occurs, which is essential to the processing and degradation of RNA. Exo9 functions to modulate RP46, 44, and RP6 RNase activity. In addition, Exo9 subunits along with RP44 help to bind phosphate groups along the RNA backbone. The topological constraints of the barrel promote strand separation of duplex RNA at the 5' end of the binding path. Essentially, the catalytically inert Exo9 organizes enzymatic activities of the exosome and facilitates substrate recruitment. Specifically in Exo9, the cap proteins function to bind the 3' strand of RNA and melt the first base pair of the RNA duplex that enters the Exo9 channel due to its structural features. RP6 functions to stabilize the exosome complex. RP6 stabilizes CS14, a cap protein, in an appropriate RNA binding conformation. This allows RP6 to indirectly contribute to the long RNA binding path. In addition, RP6 stimulates RP44 activity. There are two pathways the RNA can take through the exosome. In the more common long RNA binding path, RNA is threaded through the Exo9 central channel, which triggers RP44 to adopt a close conformation. In the close conformation, the pin domain of RP44 contacts CSD2 in the exoribonuclease region. On the other hand, the short RNA binding path allows RNA to directly access RP44, which adopts an open conformation rather than passing through Exo9 first. The video shows exonuclease domain of RP44 going from the channel conformation to the direct access conformation, which was completed with UCSF Chimera.
In summary, EXO11 is a eukaryotic nuclear exosome that is made up of the catalytically inert EXO9, the active RNase RP44, and the nuclease RP6. RP44 can adopt either a closed conformation in the long RNA binding path or an open conformation in the short RNA binding path. The EXO9 subunits bind RNA with sequence unspecific interactions reminiscent of archaeal exosomes. In addition, the channeling mechanism of exosome is highly conserved from prokaryotes to eukaryotes. The significance of exosomes as an ancient molecular machine can be seen in the presence of exosome-related complexes in bacteria, archaea, and eukaryotes. EXO11 is important for cellular function because it functions as a major 3' to 5' degradation complex in eukaryotes. EXO11 modulates the abundance of transcripts and helps to detect and eliminate defective RNA molecules. In the image on the right, it demonstrates the difference in active sites between the archaeal, bacterial, and eukaryotic barrels, even though the structure is similar in all three of the pictures.
One question for future research is why specifically is the Exo9 subunit complex necessary for RNA degradation when it has been proven that the RP44 has catalytic activity without the Exo9 complex? Another question for future research is an exosome associated with every RNA molecule but only degrades defective RNAs? The image shows the pin domain of the RP44 with a close-up view of the endonucleolytic site exposed to the solvent.
Thank you for your time.